All right, good afternoon. Today we are going to look at DNA isolation, purification, and quantification. So, um, in today's world, DNA analysis is super important, especially with real time PCR. And finding the right isolation system for your specific applications is critical for the success of your experiments. So, this lecture will give you general information on DNA extraction, plasmid preparation, and DNA quantification, and optimized purification techniques that increases productivity, allowing more time for experiments and data analysis. So, what is a basic isolation procedure? There are 5 basic steps to DNA extraction that are consistent across all purification chemistries. So, first, we have to disrupt the cellular structure and create a lysate, and then we have to separate soluble DNA from cell debris and other insoluble materials. Next, we bind the DNA of interest to a purification matrix. Then we wash proteins and other contaminants away from that matrix. And then we illusionate, we have illusion of the DNA, or we look at it. So, lyse, separate, bind, wash, elute. So, the first step, we have to create a lysate. So, the goal of lysis is to rapidly and completely disrupt the cell in a sample to release the nucleic acids into a lysate. So, the first step of any purification reaction is releasing DNA and RNA into a solution. There are 4 general techniques for lysing for lysing materials. We have the physical methods, the enzymatic methods, chemical methods, and a combination of all 3. So let's go through these 3 or 4 and look at each and every one of them. The physical methods typically involve grinding or crushing to disrupt the cell wall or tough tissues. Typically, we can see freezing and then grinding samples with a mortar and pestle. Grinders can be simple, so manual devices or automated devices, and we can see these automated devices being capable of disrupting up to 96 well plates, multiple 96 well plates. Other devices used are bead beating or shaking in the presence of a metallic or ceramic beads that physically disrupt the cell or the tissue wall. We also have scientific um, disrupt to disrupt tissues and lyses and lyse cells, so bust them open. Our chemical methods can be used alone to easy to lyse material such as tissues, cultures, um, chemically chemicals often used are going to be detergents and chenotrophs. So SDSs, guanidine salts, and alkaline solutions. And then finally, our enzymatic methods. Enzymatic methods are also often used with more structured starting materials, and the combination of other methods with tissues, plant material, bacteria, and yeast. Typical enzymatic treatments include lysosomes, zymolase, and lactase, as well as proteinase K, cholinerase, and lipase. All of these are enzymes with, as you know, with the the ending ACE. So these enzymatic treatments can be mealed by high put throughout producing. But they also have high per sample cost compared to other disruption methods. So you can get a high turnout, but it's going to cost you quite a bit more. In many protocols, a combination of chemical disruption and another is often used. So it's chemical disruption of the cell is rapid and it inactivates proteins, including nuclease. So once we have disrupted the membrane, we can clear the lysate. So we can start purifying that DNA. So, depending on the starting material, cellular lysate may have um, cellular debris to remove prior to purification to reduce the carryover of unwanted material. Usually, clearing is accomplished by centrifuging, filtration, or a bead based method. So, the important, oh, and we'll talk about these three methods next. So, centrifugation can require a more hands on time, but it is able to address large amounts of debris. So, you put your cellular debris in a tube, you set it in your centrifuge, and you let gravity do the work. So, it separates by size and weight. The heavier stuff goes to the bottom. Um, the lighter stuff goes to the top because it spins it so fast. You'll start seeing gravity pull them apart. Filtering can be a rapid method, but samples with large amounts of debris can clog a filter. And then finally, bead based clearing. We see this used quite often in automated processes, but it can overwhelmed by biomass pretty quickly. So once we have cleared the lysate, we can move on to the purification matrix. So, binding to a purification matrix, regardless of the method used to create and clear a lysate, the DNA of interest can be isolated in different methods. Each of these chemistries can influence the efficiency and purity of the isolation, and each have characteristics specific to that type of chemistry. Binding capacity is indicated by how much nucleic acid and an isolation chemistry can bind before it reaches the capacity of the system and no longer isolates any more nucleic acid. So, we're going to look at a couple of different types um, of binding purification matrices. So, we have solution based chemistry. So, following that creation of a lipase, the cell debris and proteins are protected using, or precipitated, sorry, using a high concentration salt solution. So it means it's pulled together. The high concentration of salt causes the proteins to fall out of the solution, and then we can centrifuge and separate soluble nucleic acid from the cell debris and the proteins. Um, the DNA is then precipitated by adding isopropanol uh, to the high concentrated salt solution, forcing the DNA to clump together. This forces large genomic DNA molecules out of solution while smaller RNA fragments remain soluble. The insoluble DNA is then pelleted, so squished together, and separated from the salt, isopropanol, and RNA fragments via centrifugation. This type of chemistry does not rely on a binding matrix, but rather on alcohol precipitation. So there's no binding of the DNA. We are just getting precipitants or um, things to fall out of the solution to get that DNA. Silica binding chemistry is a DNA purification system based on binding of DNA to silica under high salt conditions. The key to isolating any nucleic acid with silica is the presence of a kinetopic salt like guanine hydrochlorine. Kinetrophic salts present in high qualities can disrupt cells, deactivate nuclease, and allow nucleic acids to bind to silica. Once the genomic DNA is bound to the silica membrane, the nucleic acid is washed with a salt ethanol solution. These washes remove contaminating proteins, lipopolysaccharides, and small RNAs to increase the purity while keeping DNA bound to the silica membrane column. Binding to silica is not DNA specific, so if pure DNA is required, there is also an option to add ribonuclease or to an elution buffer. The RNA is co-purified with gDNA, and the addition of R nuclease ensures the removal of the vast majority of contaminating RNA. For automated purification, we can do 96 well silicate membrane plates to automate this and have robots do it as well. Cellulose binding chemistry. With this technology, nucleic acids bind to cellulose in the presence of a high salt and alcohols. Conditions can be adjusted to preferentially bind different species and size of nucleic acid. As a result, the combination of binding capacity and relatively small elution volumes, we can get high concentration elutions of nucleic acids. Elulates, sorry. Ion exchange chemistry is based on the interactions that occur between positively charged particles and negatively charged phosphates that are present in DNA. The DNA binds under low salt concentration, and contaminating proteins and RNAs can be washed away with a high salt concentration. The DNA is eluted under these high salt concentrations and then recovered by ethanol participation. Then we have washing, which is wash buffers generally contain alcohol that can be used to remove proteins, salts, and other contaminations from a sample or the upstream binding buffer. We can also see alcohols additionally help associate nucleic acids with their matrices. So we'll wash them over to help purify that sample. And then elution. Elution refers to the process of removing a substance that is attached to another by washing it with a solvent. DNA is soluble in low ionic strength solutions such as a TE buffer or nuclease free water. When such an aqueous buffer is applied to a silica membrane, the DNA is released from the silica and the elute is collected. The purified high quality DNA is now ready to use in a wide variety of demanding downstream applications such as multiplex PCR, coupled with in vitro transcription and translation systems, transfactions, and sequencing reactions. When selecting the buffer, it's important to consider the requirements of your downstream process. So, some downstream processes that we should know about. This is the quantification. Selecting a downstream method depends on specific research goals, such as identifying genetic variation, measuring gene expression, and sequencing the entire genome. Techniques like capillary electrophoresis, qPCR, dPCR, and various forms of NGS or next generation sequencing each offer unique advantages. Understanding the requirements for DNA input, sensitivity throughout, uh, and specific applications needed is critical for decision making processes. So, quick overview of what these things are. Capillary electrophoresis is commonly used after DNA purification for the precise analysis of DNA fragments. It is particularly well structured or suited for size based separation, making it valuable for the applications such as genotyping and DNA fingerprinting. Accurate results depend on high quality, precisely quantified DNA, ensuring reliable fragment sizing. qPCR and PCR. So, qPCR is qualitative polymerase chain reaction, and it is known for the ability to quantify measurements in the amount of DNA or RNA in a sample, making it an invaluable tool for applications like gene expression analysis, pathogen detection, and genetic testing. This technique is highly sensitive and specific, allowing quantifications of very low quantities of nucleic acids, which is essential for clinical diagnoses, research, and monitoring of disease progression and treatment efficiency. So, qPCR is often used for amplifying specific DNA segments and analyzing gene expressions or mutations. It requires DNA that is free from inhibitors and precisely quantified for accurate amplification dynamics. So we can see what that looks like here. All right, cycle numbers, fluorescent thresholds, and then we can see what qPCR, the steps are. dPCR, or digital PCR, offers exceptional precision and sensitivity in quantifying DNA, making it ideal for detecting low ambience targets, rare genetic variations, or slight changes in the gene expression level. dPCR is particularly valuable in clinical diagnoses for measuring viral load, rare mutations in cancer, and the absolute quantification without the need of external standards. Downstream analysis using dPCR provides absolute quantification of DNA to copies, which is useful for detecting low ambience targets, so targets that aren't super high. It needs high integrity DNA to avoid quantification errors. So we can see the steps to dPCR here. We have our preparation, our digitalization, amplification, and quantification. Our NGS, or next generation sequencing, is chosen for its comprehensive analysis capabilities, enabling detailed examination of the entire genome or specific genomic regions. It's ideal for uncovering gene variation, understanding complex genetic disorders, profiling gene expression, and exploring epigenetic changes. NGS can handle large sample volumes with high throughput, providing deep insight into genetic information, which is super important for advanced personal medicine, studying biodiversity, and enhancing our agricultural practices. So we can see it here: extraction, library prep, sequencing, and analysis.